Thanks, Wenjing. I may, after we've had remarks from others, come back to particularly some of the issues that were faced in 2009. So maybe you could be thinking about some examples and how they were addressed. Okay, our next speaker is Karen Midthun. She's currently serving as the Senior Advisor on Regulatory Issues to the Centre for Vaccine Innovation and Access at PATH. And before that, she spent 23 years at the US FDA, with her most recent position there being the Director for Centre for Biologics Evaluations and Research. So she has a wealth of regulatory experience, and I'm sure she's going to offer us some nuggets today. Thanks, Karen.
Thank you, and it's a pleasure to be here this afternoon. As you've heard, I was the director of the Center for Biologics and Research at the US FDA at the time of the 2009 H1N1 influenza pandemic. As you know, CBER is the center at FDA that regulates vaccines, so I'll of course be addressing things from that perspective. CBER has a long history of collaborating with the World Health Organization, both as a collaborating center for biological standardization and as an essential regulatory laboratory for influenza vaccines, and we're involved in the potency reagent issue that Wenjing was talking about a little while ago. We also work very closely with the Department of Health and Human Services here in the U.S., and with other HHS agencies, in particular the CDC and NIH, and also BARDA in vaccine-related matters. A lot of groundwork was laid before the 2009 pandemic. HHS had issued its pandemic influenza plan in 2005 with input from multiple agencies, including FDA. CBER had issued guidances on licensure of seasonal and pandemic influenza vaccines in 2007. And WHO, working together with many national regulatory agencies, including CBER, as well as other stakeholders, issued guidelines on regulatory preparedness for human pandemic influenza vaccines, also in 2007. In addition, CBER had learned lessons from the influenza vaccine shortage that the U.S. experienced in 2004, when one of 3 FDA-licensed manufacturers of seasonal influenza vaccine experienced a manufacturing problem. This led to a significant vaccine shortfall for that season. Subsequent to that, CBER used the accelerated approval pathway to license 3 additional seasonal influenza vaccines between 2005 and 2007. And this was critical to pandemic responsiveness, given that the pandemic vaccine manufacturing is built on top of the seasonal vaccine infrastructure. So instead of having 3 manufacturers, we now had 6 manufacturers. Some of the regulatory issues that occurred during the 2009 pandemic were expected because of extensive pandemic preparedness planning orchestrated by WHO. These included the need to develop reference strains and reagents for vaccine manufacture and biocontainment procedures. Some of the issues that were encountered but not necessarily expected, such as initially low yields in making the vaccine from the reassortant vaccine candidates and the difficulty in isolating purified influenza hemagglutinin to make the antiserum that is required for the potency test for the vaccine. One of the critical regulatory issues for us was to determine the most expeditious pathway to the availability of pandemic vaccines, preferably as a licensed product, although the potential for an emergency use authorization also existed. An emergency use authorization allows use of an unapproved medical product or an unapproved use of an approved medical product under certain circumstances. CBER determined that it could license the monovalent 2009 H1N1 pandemic vaccine as a strain change supplement to the manufacturer's existing licenses for seasonal influenza vaccine. That process allows new strains to be incorporated into the vaccine based on non-clinical and chemistry manufacturing control data. This is the process used every year to change the composition of the seasonal influenza vaccines that target H1N1, H3N2, and B lineage influenza viruses. Both manufacturers and the NIH conducted clinical trials of candidate 2009 pandemic H1N1 vaccines in parallel with vaccine production to evaluate the dosing regimen. Preliminary data, as well as final data from those studies, supported the usual 50 microgram dose and the adequacy of 1 dose for adults and older children and the need for 2 doses in younger children. And these clinical data were not required. We went ahead with vaccine manufacture and formulation with the assumption that this would be the case based on previous experience with H1N1 vaccines. Regulatory agencies in other countries or regions pursued somewhat different pathways, depending on their regulatory precedents. For example, the European Medicines Agency had approved what they called mock dossiers for adjuvanted H5N1 vaccines. And their most expeditious pathway to licensure was to switch out the H5N1 antigen with the H1N1 antigen, again supported by some limited data. An underpinning to the 2009 pandemic was extensive collaboration and information sharing among regulatory agencies coordinated by the WHO, as well as among CBER, other HHS agencies, and manufacturers. Other essential activities were the expanded post-marketing safety surveillance for the pandemic vaccines that had to be set up, and also communication, including with the public and expert advisory committees, was a key element as well. As you know, it took 6 months to make vaccine available, and the bulk of licensed vaccine did not become available until after the epidemic had peaked. So, what were the lessons learned? We clearly needed to more rapidly identify higher yield vaccine candidates for vaccine production. There was a need to develop potency assays that rely on reagents that can be provided more rapidly. Also, the desirability to have alternative vaccines made with newer technologies that might lead to more rapid manufacture, such as cell-based and recombinant hemagglutinin influenza vaccines. Also, adjuvanted vaccines that would provide for antigen sparing, and also, ultimately, vaccines that provide broad and durable protection against a broad swath of influenza strains. Since then, significant progress has been made with regard to accelerating the process of providing higher yield vaccine candidates for manufacture, and much work has been done toward developing alternative potency assays, but none yet that have been landed upon definitively. Also, since that time, FDA has licensed a cell-based seasonal influenza vaccine and also a recombinant hemagglutinin seasonal vaccine, an adjuvanted influenza vaccine for seasonal use, and an adjuvanted H5N1 vaccine have also been licensed since 2009. And I think, as we all know, longer term, the development of influenza vaccines that provide broad and durable protection against influenza viruses, the so-called universal influenza vaccine, would provide the best pandemic as well as seasonal preparedness. But that is still a work in progress, as we have heard. Another important development since 2009 is increasing seasonal influenza vaccine manufacturing capacity globally. In large part, BARDA has supported that as well. And that's also really critical, as we've discussed, having global capacity to manufacture, having global capacity to evaluate the issues, global regulatory capacity are all really important on an ongoing basis. WHO has its pre-qualification process for vaccines, medicines, and devices that have been licensed or approved by various national regulatory authorities. And that pre-qualification process can then allow countries, as they choose, to introduce these vaccines in a more streamlined basis. And also, WHO established a procedure for an emergency use assessment and listing in 2015 in response to the Ebola outbreak, and now has drafted a revised emergency use listing procedure and is accepting comments on that until the end of the month. And this procedure could be an important mechanism for making unapproved products, including vaccines, widely available in the setting of a pandemic. However, the ultimate decision to use an unapproved product is made at the national level, and so having the infrastructure to allow those decisions to be made and supported are really critical to the success of this going forward. Thank you.